Bring up Nirav Shah. He's our next moderator. He's the senior scholar at Stanford's Clinical Excellence Research Center. He focuses on patient safety and quality, innovation in digital health, and strategies required to transition to lower cost patient-centric care. Previously, he was the Senior Vice President and Chief Operating Officer for Clinical Operations for Kaiser Permanente in Southern California.
Thank you so much. Thank you, Rachel. It was a great talk. We're going to start with a quick audience poll here. Hope you don't mind. If you're primarily an investor, can you stand up? So we just know who to target after. No, I'm just kidding. If you think of yourselves primarily as an entrepreneur, please stand up. Okay. If you were taking a tour of the museum and accidentally wandered in here, can you please stand up? Okay, good. Well, I'm really excited about this next panel. We have three dynamic, exciting folks who are at the top of their game, and they're going to teach you and me a lot about the life sciences. I thought I knew a lot about the life sciences, but when I started talking with these guys, it was obviously I knew nothing. So we're going to change the order up a little bit. It's going to be David Charlot, the founder and CEO of Shola Biosciences. Leo Petrosian will be second, co-founder and CEO of Neural Analytics, focusing on brain health. And then we'll end with Basil Dayat, co-founder and CEO of Zencore, which is focused on immunotherapeutics. After 5 minutes each, we're going to have a Q&A session that is going to be unlike every other Q&A session. As a professor, I reserve the right to use the Socratic method and pick on anyone in the crowd to answer the question. So just be aware. And why don't we start first with David.
Cool, thank you. I guess this is the clicker here, forward, back. Oh, let me go back. Okay, well, thank you for your time. Thank you for the opportunity to be up here. I have a nice startup in San Diego. Well, actually, I started the company in San Diego, but I moved it to UCI because of a great opportunity at the Applied Innovation Center. I'm a part of an incubator called Wayfinder, and actually, in a couple of months, I'm going to be moving yet again to LA, and I'm going to be a part of UCLA's incubator called Magnify. So you could say that I'm actually touching on the major cities that people have mentioned, San Diego, Irvine, and LA with my business. And so what we're doing at Shola Biosciences is actually making the best cell analysis technology that the world needs. A lot of people talk about the value of genomics and proteomics, but that's just because we don't know how to study cells by themselves. I don't talk, when I see you and I talk to you about your day, I don't actually ask you about, you know, your bones or your toes or your nails. I ask you about all of you because you're the sum of all, not just one component. Proteins and DNA are a subcomponent, but because it's very hard to study cells, we have to actually blow them up and look at insides to kind of piece back what's happening. So we're creating a technology that allows functional cell analysis. And what I'd like to talk to you about in brief is the journey of how new technology can change the world. So, you know, the roadmap for the company, we're raising money. We're raising $3 million. Ultimately, we want to tackle clinical diagnostics and make better drugs, but in order to get there, we're going to tackle the research market and work in a few areas that are very important. Those are, of course, gene editing verification. CRISPR and other types of gene editing techniques have a huge prominence in the field. We're going to have a tool, we have a tool that helps in those areas. We're going to tackle microbiome research and as well stem cell differentiation. So a little bit more about the company. We're commercializing technology from a few universities. UCI is one. We also have technology from Arizona State University, Caltech, and technology that was developed by myself at Shola Biosciences. We incorporated in October 2016, and we have $300,000 in investment to date. $200,000 came from myself, $50K from my handsome CFO there with his hand up, and another angel investor. And big impact, why are we doing what we're doing? The power of tools really make us see things that we can't. And right now, all the drug discovery that happens in the world talks about targeting a specific pathway or something of this nature. But in reality, what cell is the pathway you're targeting in? Side effects is the biggest challenge. If you ever watch a drug commercial, you'll hear that, oh yeah, it's going to cure your diabetes, but it may also cause diabetes. That's a challenge because we don't have good ways to study side effects because we don't have good ways to study different cells. And so let me walk you through the basics. My VP of Product Marketing, Macias, who's right behind you as well with his hand up, he animated this. So this looks like a type of cell, but it's not your traditional cell model. So we spend $60 billion a year to do this. Identify the differences between two cells. How do we do it? You know, we study proteins, nucleic acids, cell morphology, or other things, and we do it for one reason, to identify a predictive signature of disease so that we can either diagnose it early so you don't need any therapy or just maybe eat more broccoli or take a multivitamin, or to figure out what type of drug therapy to give you. And so this is the idea that we have. Either we study cells out of context because we kill them, that's the only way we can get to a genomics of protein, it's inside the cell, or we use messy and confusing labeling strategies where we actually apply a tag to a cell because we think we have a specific signature of that cell, or we study them in a happy way. And that's what we do at Shola Biosciences, is we use a proprietary technique that we have out of ASU called self-phoresis. Thinking about branding strategy, I was mentioned, we are able to isolate and separate different types of cells as you would DNA in a gel. So we have self-phoresis. Competitive landscape, you know, there's forensic science style techniques where forensic science, you're studying a dead thing, DNA and proteins, or in the life science aspects, you have different methodologies. We have what we're creating called functional live cell sequencing. It's a new buzzword. A lot of people, when I say it, they're saying, what do you mean, how can you sequence a cell? Cells are all different. We have technology that enables that. So our technology, people talk about things in three. These are the values we have for our technology. One, we can quantify the spread of cells. They all look alike, and so a lot of people, when they do cell research, they start with a monoclonal population of cells. It's supposed to be the same population, but there's still a spread. We're able to identify that spread and allow even better selection of the right group so that as you're doing your development, you don't have variability, which is the biggest challenge. And lastly, for gene editing very specifically, we can help validate that gene editing actually took place and worked at a low price point. Here's our instrument. We have a printer ink kind of model, so we have an instrument and a consumable. And in terms of customer traction, we have multiple use cases. And so gene editing is going to be our first one because of the problems in CRISPR gene editing, which people talk about all the time. And we also have a collaborator at UCI who uses our technology for studying neuronal stem cells, how they're transitioning from a pluripotent stem cell to a faded neuronal cell. The IP, as mentioned, comes from Caltech, ASU, UCI, and stuff that was developed at Shola Biosciences. You know, what we're raising is $3 million to actually accomplish the first part. New technology, you have to take care of the infrastructure. It's not actually super sexy to investors, but it's required. And I guess I'm a little over time, so I'll just get to the final goal. We sell into the marketplace, make a lot of money, and have a great ROI for investors. Who powers this operation? It's the team. Myself and CFO, we take care of corporate governance. In terms of our science, we have a great CSO who understands cell biology, CTO who understands microfluidics, and great sales team that understands how to deploy new technology into the marketplace. And in conclusion, we have the 3 T's: new technology, a great team, and the timing. CRISPR editing, it's poised to change the world, or genetic engineering. Here's a label from a Campbell's tomato soup can. Everybody says, oh, GMO versus organic. Those buzzwords don't actually matter. All foods are going to be genetically engineered in the future. The USDA has actually said that they're not going to even regulate it. They gave a cute reason why not. It's too expensive to regulate is why. We have a tool that actually helped them regulate it. So thank you for your time. Sorry for going over a little bit.
Leo, you're up.
So I'll get my slide. No slides for today. I'll be brief. So ultrasound imaging is a very valuable and versatile imaging modality that's used for thousands of different applications, millions of times a day, all over the world. Though it's been around since the 1950s, and there's seen over the last 70 years tremendous amounts of innovation in the technology, the entire industry has overlooked 2 of the largest bottlenecks in all ultrasound imaging. Is that if I was to give this gentleman here an ultrasound machine and show him an unconscious patient, and I'd say, is this patient okay? He'd look at me and he'd say, Leo, I don't know how to use this machine, and even if I could figure it out, I wouldn't know what it's telling me. So what we do at Neural Analytics is that we take known ultrasound modalities, known ultrasound value propositions. For example, an echocardiogram can tell me about the health of somebody's heart. That's a known practice. And instead of having a technician operate the system and a physician interpret it, what we do is we substitute a fully autonomous robot for the technician portion and AI or machine learning derived algorithms for the physician's portion. So at the end of the day, what you can have is a fully autonomous robotic ultrasound system. The benefit that this enables is that you can now take ultrasound that's historically been confined to places like hospitals And you can make it available in an ambulance, in an urgent care, in a nursing home, in combat, in the battlefield, in a submarine, or in many, many other environments where you do not have the operators or interpreters. Most acutely, we work in the field of brain health. Why brain health? Brain health accounts for approximately 4.4% of US GDP. 1 out of every $20 in America gets spent on brain health. I find that number astoundingly high, right? $760 billion a year is estimated by the CDC, of which $343 billion is attributable to 3 diseases: dementia, stroke, and traumatic brain injury. The problem with these conditions is that, like all other aspects of healthcare, healthcare is a fundamental relationship between a diagnostic and a therapeutic. In those conditions, there's really no good way to rapidly assess the health of the brain. What we've done is we've taken known ultrasound methods for assessing these diseases, and we've made them fully automated, now available in other clinical environments. Our first product, which we've been working on for 5 years, is a fully autonomous robotic ultrasound that could rapidly determine if a patient has suffered a stroke. And actually, after 5 years, this product obtained European market clearance last Thursday and US market clearance via the FDA last Tuesday. So we've had a pretty busy week. After our first product, which is designed for rapid stroke detection in the field, we also developed tools for rapid brain injury detection in partnership with the United States Army. The Army gave us a development contract to develop a field-based system for rapidly assessing the health of soldiers' brains once they have a trauma in the field. And we're hoping to launch that product late next year. A little bit about the company. We spun out of UCLA Neurosurgery around 5 years ago. We've raised approximately $45 million. We've won $16 million in government grants and contracts. We have about 95 employees to date, about a third of the company's PhD engineer scientists. What we're working to do is usher in a new age of ultrasound imaging to change the paradigm of healthcare, and I'm here to answer any questions that you might have. Thank you.
Basil?
Hi, I'm Basil Dahiat. I'm CEO at Zencore. We are a biopharmaceutical company based in Monrovia, which is a few miles from Pasadena. And we spun out from Caltech in 1997, back in the Stone Age when Stefano and I went to Caltech together. It was notable when he said that he could now retire from UCLA and be an emeritus professor, which is fantastic. When CEOs change jobs, it's called being fired. So I think that's something we should always keep in mind when we're in the business side of things instead of in cozy academia. So what we do at Zencore is we engineer molecules that are called monoclonal antibodies. They're a component of your immune system that evolved to recognize the myriad of different pathogens and other insults your body faces. And so what we've done is we've found a way to manipulate them to optimize their properties, to optimize their drug-like properties. We're currently running programs. We have 10 programs that are in clinical testing, 5 with partners, 5 of our own. I'll tell you about our business model in a second. We're about 140 people. We opened a second site in San Diego about 3 years ago. We are listed on NASDAQ. We have about a $2 billion market cap. So if you want to invest in an exciting Southern California biotech company, just take out your E-Trade, type in XNCR, and buy. Now, back to the founding story. So in 1997, I finished my PhD, and I was at Caltech, and it's the greatest place in the world for research, I will say with no reservations, and all the MIT people can kind of yell at me. What we did was we developed algorithms, high-performance computing algorithms, high performance at the time, to manipulate the amino acid sequences of proteins and make them do things better. And don't even ask me what we were doing back then. Caltech was fantastic for this. Obviously, amazing technology, amazing people I got to work with. The tech transfer office was incredibly constructive, and I heard it's even better today. So these are places that want to get guys like you to invest in the technology coming out of them. We got great investors initially, a family office, but unfortunately they're not from LA, they're out of Chicago, right? That's a common theme. There's so much less competition in LA, you can go there and really have an impact. So we sort of muddled along, trying to figure out what to do, working, doing our little design work for ag biotech companies like Novartis Seeds and Dow Chemical for industrial transformations, and then went out to raise money in 2000 and accidentally raised $50 million in a private financing because it was the dot-com boom. So thank you, Silicon Valley. That changed Zencore's fate. That was our first big stroke of luck, and luck is, of course, the most important ingredient in anything we do. So we raised that money, and we started working on therapeutics. We sort of got ourselves into the monoclonal antibody game by realizing that the bottom of the antibody, this little modular pop-up unit, could be optimized to do all sorts of things and then pop back on. And so you have like almost a software model for discovery and partnering. We optimize them for longer half-life, so you can go from twice-a-week injections to every 2 months maybe. Optimize them to kill cancer cells. We optimize them to clear out antigens better. And these are all now in our clinical programs and our partners' clinical programs. We expect our first drug approval in the fall, our partner Alexion, for a rare blood disorder that causes paroxysmal spasms at night and also potential death. So for us, though, developing our own drugs really got going after sort of about 2005. We brought in a venture round. We started our first clinical trial. And that's when everything really changed for me and for the company. We really realized our true mission and our true focus. Our first clinical trial was in relapsed Hodgkin lymphoma, disease with a 90% cure rate, 10% fatal in about 2 years. We got a patient, a 39-year-old woman at MD Anderson. She was the 16th patient in our trial, so we'll call her 16. We never know their names. She had a near complete response of her tumor. It almost melted away. It was an amazing time for us because by chance in 2009 when that happened, we were out of money. The markets were dead. That led us to a $7 million convertible note with our insiders and go on. She went to Mexico to visit her family, saw her family again, and then came back and died a few months later. And the guilt that we carry with us for moving on when she passed away is really only bridged by all the impacts we've been able to take and make from that $7 million we got because of her contribution. So now, like I said, there's 10 clinical programs. The second one ought to be approved sometime next year. We are dedicated to investing in our own programs. We're going to start our sixth trial of an internal program, our sixth program with a trial. We've run many, many trials later this fall. And I was just at a patient conference for neuroendocrine tumors, interesting Silicon Valley link, that's the one that killed Steve Jobs. And I got to meet with patients yet again. And every time I do this, I go away with the feeling like we're not doing enough. So the kind of technologies that we have in LA that people with the capital and expertise in Silicon Valley could really bring forward and make a real impact for human well-being and ease suffering, I think they're just waiting for you guys. So I really look forward to welcoming you to LA.
Thank you so much. So we're going to run this a little differently. If you have burning questions that you would like to ask of the panel, write down your name, write down your question, and hand it to me, and I may or may not choose your question, depending on what's attached to that. There's a lot of wisdom in this room as well, and while I want to have the panelists get first crack at answering the questions, if there is anyone who has a burning desire to respond to one of these questions, please just come up to the side and we'll have you speak as well. So I'll start with my first question, and these are hard questions. They've not heard them before. They have no idea what to expect. So Scott Galloway of NYU Stern writes that in the digital world we live in, what matters are analog moats. So for example, Amazon building these multi-trillion-dollar warehouses everywhere protects them strategically from everyone else because no one can enter the market unless they build all those warehouses as well. In the life sciences, some of those analog moats are the FDA, the insurance companies and what they choose to pay for or not, physician practices, whether they allow your ultrasound to be done by a tech or they monopolize it, things like that. How are you getting past some of these analog moats that you've encountered? And you don't have to, if you have no answer, that's okay. It's not 1, 2, 3. It's.
Is it okay if I chime in? So what we opted to do is not try to get past the analog moats, but to understand where they all are and leverage all of them, right? So this gentleman over here started a company 21 years ago, and he's going through a process of building something completely revolutionary and new. We opted, we're slightly much more impatient, I should say. So what we said was, what can we do at the speed of a tech company in life sciences? And so that's how we ended up doing exactly what we do, is we take known technology that has already superseded these moats. meaning like we use existing reimbursement, existing regulatory, existing clinical guidelines, existing standard of care. But then we create our own moats right behind us. So as an example, like our system works off of AI on robots doing ultrasound. Protection of IP around AI control systems or whatnot, that's really challenging. So what we do is we have a tremendous amount of IP on robots doing ultrasound. So that is a moat we left behind us. So if anybody wanted to come in and compete with us, they'd run right into our robots doing ultrasound IP portfolio, right? So what we opted to do is basically understand what was the quickest meandering path to market, like we can think like a tech company pursuing an MVP, and then what are the moats that we could drop behind us once we go through it.
Gotcha. Well, for my company, what we're doing is actually first tackling the research market, so we don't have these moats to have to deal with in terms of regulatory. I like this. So my previous company, I showed it briefly up there. I worked at a company called Biological Dynamics. It's a startup company in San Diego. Started out of graduate school with my buddy, and it's oncology diagnostics. So we took great UCSD IP, and we ran right to the FDA saying, we're going to change oncology practice. We have a marker that shows up in blood that we can diagnose disease early. And what I learned through that process, I was CTO and president of the company for 6 years, you should actually do what Illumina did. Go to the research market, have it proven, have lots of publications, have the confidence of the world that the technology works and it can be trusted, and then go into the FDA. So that's a strategy that we're deploying at Shola Biosciences. We can displace existing hardware that's known and used because it's better, faster, cheaper, and easier to implement. But instead of running directly to the FDA, we're going to research markets because that's where you're going to get the best actual outcome, because we're going to be able to make new tests and research that can then translate into the clinical space.
That's great.
So I would say the biggest moat in biopharmaceuticals is the massive amounts of cash that you need to discover, develop drugs, and then even larger to market and distribute them. So the approach we've taken is really try to steal those moats and use them ourselves by having the best technology that can be readily applied in many different programs, and choosing some of them to sell, and others where, because of the leverage we have, if we can really be, just have enough foresight to have the best technology that'll fit in the moment, and do partnerships where the large company kind of has to come to it on our terms. So our partnership with Novartis we did about 2 years ago for a new set of hematologic malignancy drugs. We kept U.S. commercial rights, but they have to pay half of the costs. We kept 70% of the value. So if you can have a technology that is just timely enough, then you can really invert that moat and use it for yourself.
Nice. Very nice.
But it took about, 20 years.
Yeah. Any rebuttals from the audience or any other comments or thoughts? I'm offering. You're welcome to take me up on the offer. My next question is regards to talent. And there's a lot of myths about talent. One of the myths I've heard is that Stanford grads hang around long enough to learn, and then a year later, they have to go off and do their own startup. What are the myths about talent in Southern California that you've encountered?
That there's not a lot of technology talent in Southern California. I would say there's immense technology talent in Southern California. And it is the case that the dispersedness of the ecosystem there makes it harder for people to move around. So it's the truth, you have much lower turnover. We have staggeringly low turnover in our home base in LA, and our San Diego site is significantly higher. Yeah. Right? So I'd say that's a total myth.
Why?
What?
In San Diego?
Because it's a life sciences company, there's a life sciences cluster, and they're all within about, 15 miles?
Got it. So Southern San Diego's more like Northern Cal?
In that one way within life sciences, I suppose.
Fair enough.
It really has to do, it's always about a pursuit. You have two resources that we're leveraging always, capital and talent, okay? If there's an abundance of capital, there's going to be a dearth of talent because there's so many people vying for it. If he's in an environment where there's a number of bio companies all doing drugs, then they all have a lot of, lots and lots of money, they're going to be vying for the same talent. We have the good fortune of, we opted to spin out of UCLA, and we happen to be probably one of the very, very few companies that's doing what we do, robots, AI, ML, and healthcare. What that means is that we get talent of the highest caliber at a relatively very low cost. Page 8 of that BCG report on the bottom actually articulates what the cost differential is. And not only do we get them at a much lower cost than you can at NorCal, but we also keep them. We had 0% turnover for the first 4 years, then we had one person leave to go to grad school, right? And I'm still pissed off about it.
I would say a myth that people talk about is actually, it's related, but it's work ethic. So you heard two gentlemen in the last panel, they mentioned we're right in Santa Monica. We have the Pacific Ocean right at our feet. We, after a heavy session of working, we just take a dip in the ocean and then go right back to work. I saw some faces when they said that, and it was, okay, those guys aren't actually taking their job seriously. It may just, literally because they're doing that, take it longer for them to accomplish whatever they're doing. And if you're in a much more cold place like Boston or Minneapolis, or it's not as cold in Northern California, but I would say, I've been in San Francisco. I definitely appreciate Southern California more. The work ethic that's ascribed to the region, because you don't have the same level of paradise, as one would say, makes it seem like people aren't as serious as working down in San Diego. And I'll say that I'm not from Southern California. My dad was in the military. I moved in lots of places. I'm a transplant to San Diego for graduate school, and it is because of my work ethic that I stayed, because of opportunity. And it is because of opportunity that my work ethic thrived, and it's keeping going. That's why I started this company, Shola Biosciences, because there's opportunity for people who have the ambition to thrive. And so I would say that's one myth that should be overcome. It's not real.
It's also a myth that millennials don't work hard.
That's right.
We have a company full of people under 35, and they are very hungry to establish themselves, build a career. Because they're looking around and they're like, oh my God, I'm going to have to be really successful just to buy a house.
Now, if I could just provide a little rebuttal to that. They work really, really hard, but they have zero patience. That's been my experience.
Well, not to prove that, I just found out because of a change in the definition that I'm a millennial. I was born in 1981, which used to not be a millennial, but now 1980 is the cutoff, so I am. So,
So do you work hard?
Maybe I do, maybe I don't. I'm just kidding. I definitely work hard.
So I'm coming to the end of my questions, and I'm going to be picking on Andy and Steve and others who I've heard have really good questions soon. So be ready. One of the mantras I've heard now in healthcare is that we all need to be patient-centered. Patient-centered care is the new thing, right? We expect patients no longer to enter our worlds, the hospital, the clinic, but we must enter their worlds, whatever they may be, the workplace or home. And one of the ways, how are you thinking about being more patient-centered, whether it's direct-to-consumer or other strategies to become more patient-centered?
I'll go for it. So for us as a drug discovery and development company, it's about, how do you make your drug actually deliver patient-relevant outcomes? So the acronym is PCO, patient-centric outcomes. An outcome is a measurable in a clinical trial that demonstrates the efficacy of your drug, right? It's what actually goes on your product label. It's what the FDA decides, yes, you did or no, you didn't. They have a whole initiative now to try to encourage the development of patient-centric outcomes. They're trying to engage with patient groups. I will say it's going to be a long while, because these are very difficult things to quantify, right? Something as simple as, and well established in the medical practice world, that for inflammatory and autoimmune disorders, steroids, ancient drugs, are highly effective and highly toxic, and they make patients feel horrible.
Yeah.
Hot flashes, you can't sleep, your face bloats, you get diabetes, you get osteoporosis, and yet it's almost impossible to convince the FDA that just avoidance of steroids is a metric, much less to say avoidance of symptoms that are patient-reported is a metric that you can get a drug approved on. They know it, but they're struggling for the quantitative tools and for the validation to get there. So I think everybody's going that way. It's going to be hard.
If I could just follow right up on that. So we're trying to develop those quantitative tools, right? So today, when people are just designing clinical trials, they're macro-scale population-based.
Oh, yeah.
With one thing that we think is that healthcare should not be that macro-scale, those lumped together population-based systems. Ultrasound, the benefit that it has is it's completely non-invasive, it's safe, it's portable, you can put on a baby for 24 hours. So for example, why we won the Army contract was we did a clinical study around brain injury and brain injury recovery. we were able to literally go to the home of people the day that they got a brain injury and scan their brain every single day for the next 90 days. And that quantification of the recovery profile allowed us to show what are the things that influence and can accelerate the recovery. But it was the fact that what we're trying to do is make it more patient effective by getting data much quicker, because there's the partnership in all healthcare, which is assessment and intervention are intertwined. The better we assess, the better we can intervene, right?
Yeah, that patient segmentation down to the smallest unit, understanding it and then allowing tailored therapeutics is the future. We talk about it as personalized medicine at the genetic level, but really it's not even just down at the genetic level.
That's right. And for me, it's, I guess they're mentioning essentially patient stratification. If you're getting a drug to the market, it's best that if you're going to have, 100 people, that all 100 can respond versus 100 people where 7% respond or something like that. So with what we're doing in our strategy at Shola Biosciences, we're more in the basic front end. So we're really in the life science versus in the clinical diagnostic or clinical intervention stage. We're helping to craft, because of new instrumentation, we can make better metrics. We can measure better things that could actually become the clinical endpoints in clinical trials that actually drive towards these personalized medicine. Because we still don't have them, and that's why it's going to take a long time. It's not because there's not goodwill and interest. It's because we don't have the measuring tools to make those decisions. Biology is still very, very complicated.
So, Alex, next to you, the gentleman has a question. If you could either read the question or you can share your mic with him.
I can shout now. This question is for Leo. So, in your fundraising journey, you talked a little bit about where you have found successful fundraising and what challenges you've had being a Southern California company raising money.
So med device or diagnostics or imaging is like literally on the life sciences totem pole, the very, very bottom. Like at the midpoint and up is novel therapeutics. Then it's like the generic therapeutics or therapeutic devices comes next. And then way, way, way at the bottom, you have diagnostics and imaging technologies. Despite the fact that diagnostic and imaging technologies are the cornerstone of healthcare, they don't get much attention. So we started the company 5 years ago, and we've managed to raise $45 million over this period of time and win a bunch of government grants, but it has not been trivial. Alan shared a story where Qualcomm was his A, his B, and then SoftBank was his C, boom, boom, done, right? For us, we have 128 shareholders. We began with a very, very large Series seed with angel investors. Then we literally pitched every single person. So we began life thinking like, look, there's robots and AI VCs, and there's life sciences VCs, and you put them together, that's a lot of VCs. Then you're like, no, that's not true. It's like, we do robots, AI, and life sciences, and there's like 3 people who will talk to you. So we've raised money from angels in LA, VCs in China, hedge funds in Chicago, VC funds in New York, and it wasn't until this last round where a VC fund in LA actually turned around and goes, that's exactly what our investment thesis is. The intersection of robots, AI, and healthcare. And we're like, where were you 5 years ago? And they're like, the fund's 6 months old. And we're like, well, good luck for us. But it was definitely not a smooth process. At least I used to think like I do fundraising and then technology and other stuff. But for 5 years, all I do is fundraise. How do we manage all the folks on the cap table? MailChimp? So we have really, really good angels. The Tech Coast Angels, which is, I believe, the largest angel organization in LA by deal size or whatever, they led our seed round. They've been a fantastic partner with us. We have representation from them on our board. We throw parties once every 6 months and bring everybody up to speed and send out shareholder updates via MailChimp once a quarter. I got to send the most fun update of my entire life yesterday night because of the FDA clearance, which literally we pitched 5 years ago. Thank you.
Wonderful.
Yeah. 33 bottles of champagne, that's all I'll say. Yeah.
I know Steve has a good question, if you want to shout it out. Come on, you had it before, you forgot? Or Andy, either one.
sense is there isn't a lot going on. And maybe can you talk about why the life sciences cluster is not getting any local funding?
May I?
You please.
So the VC that led our B round had a really, really good quote that I love. And he said, 3 to 5 years ago, the industry was made up of a bunch of verticals. There's the automotive sector, the energy sector, life sciences, tech, e-commerce, whatever, right? And he's like, we think 2 to 3 years ago, tech fell over. It's a horizontal. And what their investment thesis is that we invest at the intersection of tech with all of the other verticals, okay? So though there's not been a number of VCs popping up and saying we're life science VCs, what we have been seeing more of is people that are looking at the intersection of tech with other industry areas. And that's what I can, with confidence, say that I've seen different groups have shown up and say, what we're interested in is how you leverage tech to change paradigms of care in other disciplines. So it's like leveraging tech to teach a industrial strength Roomba to clean floors. That is a really, really interesting application of that, right?
Yeah, I would say that there is a bit of a dearth of local money that's life sciences sophisticated. It was that way 20 years ago when I started Zencore. Our first money was out of Chicago. Then our next set of tranches were out of pharmaceutical companies based in Copenhagen and in Gaithersburg, Maryland. I'd say there's I think a bit of a cracking of the ice now. And an example of that is one of the prominent partners in life sciences at Kleiner Perkins, Beth Seidenberg, left there, starting her own fund. She will raise money like that, and she's going to base it out of Westlake Village, right next to Thousand Oaks. So I think there's starting to be a crack in the ice, and I think it's also helped by the sources that Rich mentioned, the vast amounts of money that hedge funds and whatnot in New York are now willing to deploy privately because their level of technical sophistication in the last 15 years has exploded. Yeah. They're so scientifically literate now. It's amazing. When I go to New York, I might as well be going to a scientific meeting. And this is pitching hedge funds. Yeah. So I think that the Asian money is something that I've not personally experienced except in dribbles. These are Asian public company funds that are now asking for meetings. And I'm like, who's that? Oh, we're the largest insurance company in China, and we have a $14 billion fund. We're going to do half in healthcare. $14 billion, half in healthcare? When can I meet? So these dynamics are changing, but it is starting from a low base, which again goes to investors in this area. You don't have a lot of competition yet.
And I would say that in life science, so a lot of people when they talk about life science, and the panel, its title is life science, but in reality, there's therapeutics, there is imaging diagnostics, and there's basic science instrumentation. So this is all in one category, which it actually should not be in one category, because at least for what I do in terms of basic science, although the end goal is in therapeutics and diagnostics, when people hear my pitch, they said, oh, come back to us. We know you're going to be successful. Come back to us in your Series B when you're focusing on the diagnostic. And I always say, oh, thank you for the confidence, but how am I going to get there if I don't have investment? So the phrase is, the closer you are to the university in terms of discovery, the more you need to rely on the government. But the problem with this concept is you've already relied on the government to get the discovery. That's government grants, R01s. That's how the professors pay the graduate student or what have you to develop it. And so now when you need the investment for getting it to the next stage, the investors that I've talked to, they've said, oh, that sounds wonderful. Show us all of this product adoption in the marketplace, because then we know that it has traction, it can scale, and yada, yada. But then that's the equivalent of me just going to a bank and saying, hey, I got a great idea, and the banker's going to say, prove it, and say, well, I already have customers who want to buy it, I have customers who've bought it, and I can just get an interest loan and pay it back with sales versus, take on equity. So the challenge for life science on the more basic side is there's really nowhere to go unless you write a grant, and that's kind of the biggest challenge in San Diego, because there are a lot of people like me who are entrepreneurial in spirit. They got their PhD, and they want to start a company, but then when they try, it's like, oh, I need that data, but wait, this is not even enough?
I think be very careful about getting caught on that endless treadmill of trying to feed a small company by writing grants. Yeah. You're constantly writing grants. They're small. The performance obligations to the government agencies are onerous and pointless. Yeah. And there's a rigidity to how you can use the capital that really constrains, the pivots you need to make as a company. it's tough to feed a family on a grant if you're a biotech company or any kind of life sciences.
But think of it as a stepping stone.
It's a stepping stone,
Right.
but boy, it's not much easier to get off of that treadmill as it is to try to.
Unquestionably.
You should always be trying to raise money from every possible source you can do. That I'm with, but
Yeah.
the grant thing, it's not a panacea to say a government agency just dumped $100 million in grant capital. That's not a panacea.
I just got a PhD in life sciences from you guys, so I'm going to ask for more. In the last minute we have left, one pearl that you can give this audience from your experiences that will say, I didn't think about that.
Oh, well, for me, it comes from my family, so I'm going to go left and then go right for the point. My wife, she's from Ethiopia. We have 2 babies, and my son, and we're teaching him both English and her language, Amharic, so I'm inadvertently learning Amharic. I learned about the characters and all this stuff, and it's very, very complicated. Now I'm going to get to the biology relevance. DNA, we understand the nucleotides. There are 4 of them. RNA, there's a replacement, a T to a U, uracil. It turns out all cells have these basic elements in them, but they're so different. And I would contend that what we're going to learn as technology gets better is cells, they speak different languages. It's the same characters, but it's like if you were to talk to somebody in French and they write down, if you speak English, you can read the letters, but you may not understand the words. That's going to be a big breakthrough in the genomics community, is a realization that maybe a lot of the regions that we think are not actually encoding for anything, it is because that the language that you're translating it into is the wrong language. So I'll leave you with that.
That's okay.
I'll be brief. I'll say that in probably less than 20 years, the concept of getting a second opinion will disappear in healthcare. Today, if you see a doctor, and this person could be, I got UCSF Medical School, Mass General Residency, Mass General Fellowship, whatever it might be, and you see this doctor, and if they don't tell you something you want to hear, you're like, I don't believe you, you're wrong, I'm going to go find somebody who can tell me something else. That's the standard of care today. And I firmly believe in 10, 15, 20 years, the concept of needing or wanting or desiring a second opinion will vanish. And when we go see a doctor, the doctor will not just be taking their best guess with limited data and limited information. They'll be analyzing, interpreting much more detailed, much more quantified, much more comprehensive data sets. And whether you see one doctor or another, they'll tell you the exact same thing.
I would say that the patient is rising. I think control of how healthcare is developed and how it's delivered is going to shift to patients in a fundamental way in the next few decades. I think whether it's patient-reported outcomes or centric outcomes that makes drugs relevant to them, whether it's the ability to sort of manage the crazy pricing that healthcare delivery hospital systems and pharmaceutical companies like us charge, that's all going to be driven by patients. I think they're going to demand therapies that are relevant. They're going to demand therapies that are affordable. And I think all the political ballyhoo right now is just a tiny little prelude to what happens when the patients really make their voices heard.
Thank you so much. Thanks the panel for me. Thank you.